Today, we call it neuroplasticity, the ability of the nervous system to change through experience. It sounds like a modern idea, but the search that led to it began more than 2,000 years before modern neuroscience existed. For centuries, people could see the result. Experience changed behavior. Practice changed skill. Repeated experiences created associations. But the change itself remained hidden. Aristotle had described how repeated experience could shape behavior. Centuries later, William James proposed something more radical. Perhaps the nervous system itself possessed a physical property he called plasticity. Experience might actually modify it. But that was still an idea. Scientists needed evidence. They needed to control an experience, measure what happened, and begin tracing the change into the nervous system itself. By the beginning of the 20th century, that opportunity appeared somewhere unexpected, not in research designed to study neuroplasticity. It appeared during experiments on digestion, and the scientist conducting them had been investigating an entirely different biological problem. His name was Ivan Pavlov. This is part 2 of the history of neuroplasticity, and our investigation begins where part 1 ended. Ivan Pavlov was born in 1849 in Ryazan, Russia. As a young man, he left theological study and turned towards science, eventually becoming a physiologist. His great scientific interest was digestion. Pavlov and his laboratory developed methods for observing digestive secretions while physiological processes were taking place. The work eventually earned him the 1904 Nobel Prize in Physiology or Medicine. But during those investigations, something unusual kept appearing. Food naturally produced salivation. Yet sometimes the dogs began salivating. Before the food arrived, events associated with feeding could begin producing the response. Pavlov turned that observation into a controlled investigation. A signal could be presented. Food could follow. The sequence could be repeated. And salivation could be measured. Eventually, a signal that originally did not produce the food-related response could trigger salivation on its own. Experience had changed what produced the physiological response. That was enormously important. Scientists could now control the experience and measure the resulting change in behavior. But the experiment exposed another problem. Pavlov could observe the signal. He could measure the response. What he could not yet identify was what experience had changed inside the nervous system to create that new relationship. So the investigation moved deeper. And one of the scientists who carried it forward was a young Polish researcher named Jerzy Konorski. The next important figure in this story was born in Poland in 1903. His name was Jerzy Konorski. Konorski entered science when Pavlov's conditioned reflex experiments were already changing the study of learning. As a young researcher, he became fascinated by that work. But Konorski and his colleague Stefan Miller began asking questions that went beyond the conditioned responses Pavlov had measured. They wanted to understand how learned behavior was organized inside the nervous system. During the 1930s, Konorski even spent time working in Pavlov's laboratory in Leningrad. But his scientific career was disrupted by the Second World War. After years working outside Poland, Konorski eventually returned home and continued developing a physiological explanation of learning. Then, in 1948, he published an important book, Conditioned Reflexes and Neuron Organization. The title captured where the investigation was heading. Scientists had begun with observable responses. Konorski was now asking how learning might alter the organization of neurons themselves. He proposed that learning could involve changes in the functional connections between nerve cells. Experience might therefore change more than behavior. It might change how parts of the nervous system communicated. But how could experience actually change a neural connection? At almost exactly the same time, a Canadian psychologist was developing an answer. His name was Donald Hebb. Donald Olding Hebb was born in Nova Scotia, Canada, in 1904. His path into brain science was unusual. He studied literature and philosophy, worked as a teacher, and eventually turned toward psychology. His research brought him into the growing effort to connect behavior with the physical brain. By the 1940s, scientists faced a difficult problem. Pavlov had demonstrated that experience could change a response. Konorski had proposed that learning could involve changes in neural organization. But how could activity actually alter a connection between neurons? In 1949, Hebb published The Organization of Behavior. Inside it, he proposed a biological mechanism. if activity in one neuron repeatedly helped activate another neuron. Some process could make that relationship more effective. In other words, repeated patterns of neural activity might strengthen communication between particular neurons. Today, Hebb's idea is often summarized as neurons that fire together, wire together. But those were not Hebb's original words. The phrase came later. What mattered was the idea behind it. Learning might leave a lasting effect because neural activity could change the effectiveness of the connections themselves. It was a powerful explanation. But in 1949, it was still a theory. Backhand index pointing right, could scientists actually measure a neural connection becoming persistently stronger? That would require another kind of experiment. Hebb had proposed something scientists could eventually test. If repeated patterns of neural activity strengthened communication between neurons, then the effect shouldn't disappear the moment the activity stopped. Something should remain changed. But demonstrating that required scientists to do something difficult. They needed to activate a neural pathway, measure its response, and then determine whether previous activity changed how strongly that pathway responded later. One researcher who became fascinated by this problem was Norwegian physiologist Terje Lømo. In the 1960s, Lømo was working in Per Anderson's laboratory in Oslo, studying electrical activity in the hippocampal formation. The hippocampal formation was especially interesting because of its growing connection with memory. Lømo stimulated a pathway carrying signals toward a region called the dentate gyrus. Then he recorded the electrical response produced by neurons there. During this work, he noticed something unexpected. After certain patterns of repeated stimulation, the pathway didn't simply return to its previous state. Its subsequent response could become stronger, and that stronger response could remain. This was precisely the kind of lasting physiological change scientists had been searching for. But an observation was only the beginning. Could the effect be produced systematically? Could it be measured? And most importantly, how long would the change last? Another scientist was about to join Lømo in answering those questions. His name was Timothy Bliss. In 1968, British neuroscientist Timothy Bliss arrived in Oslo and began working with Terje Lømo. Together, they investigated the unusual strengthening Lømo had observed. Their experiments focused on a pathway entering the hippocampal formation. They stimulated the perforant pathway and recorded responses from neurons in the dentate gyrus. First, they could establish how strongly the pathway normally responded. Then they delivered brief periods of stronger, high-frequency stimulation. Afterward, they tested the pathway again, and something had changed. The same pathway could now produce a larger response. More importantly, the stimulation had ended, but the stronger response had not. In experiments eventually reported in 1973, the enhanced response could persist from about 30 minutes to as long as 10 hours. The nervous system had retained a measurable effect of its previous activity. This persistent strengthening became known as long-term potentiation, LTP. For the first time in our story, scientists could experimentally produce and measure a lasting change in neural communication. Hebb's general idea suddenly had something remarkable beside it: physiological evidence that neural connections could remain changed after activity. But there was still a crucial problem. Bliss and Lømo had produced the effect with electrical stimulation. Backhand index pointing right, was anything like this happening when an organism actually learned something? That question takes us to Eric Kandel. While Bliss and Lømo were investigating lasting changes in neural responses, another line of research was approaching the problem from the opposite direction. Instead of beginning with electrical stimulation, Eric Kandel began with learning. Kandel was born in Vienna in 1929. His family fled Austria after the Nazi annexation, and he arrived in the United States as a child. He later studied medicine and became fascinated by one of biology's most difficult questions: how does experience become memory? Kandel wanted an experimental system simple enough that researchers could follow learning from a change in behavior, all the way down to individual nerve cells. His laboratory studied simple forms of learning, including habituation and sensitization. Habituation occurs when repeated exposure to a harmless stimulus causes the response to become weaker. Sensitization works differently. After a significant stimulus, later responses can become stronger. Researchers could observe these behavioral changes and then investigate the neural circuits producing them. That was the crucial step. As behavior changed through experience, scientists found corresponding changes in the effectiveness of communication at particular synapses. A synapse is the junction where one neuron communicates with another cell. Learning was no longer visible only as changed behavior. Scientists could begin connecting the learned response with measurable changes in synaptic communication. The search had reached the connection between neurons. But another question remained. Some learned changes disappear quickly. Others can remain for days, years, even decades. Backhand index pointing right. What makes a neural change last long enough to become long-term memory? Kandel and his colleagues continued following the process. And as they compared shorter-lasting learning with longer-lasting memory, an important difference began to emerge. A short-lived change could modify communication using machinery that was already present at the synapse, that could alter how strongly neurons communicated, without requiring the cell to build an entirely new structure. But when learning was repeated and the resulting memory persisted, the biology went further. Signals generated at the synapse could reach the neuron's nucleus, where genetic instructions are stored. Specific genes could become activated. New proteins could be produced. And those molecular processes could help support longer-lasting changes in synaptic connections. With sufficiently persistent learning, researchers could observe structural remodeling of synaptic connections as well. This was a profound development in the investigation. Experience wasn't simply producing a temporary response. Under the right conditions, it could initiate biological processes that helped maintain changes in neural communication over time. The question that had followed scientists from behavior to neurons to synapses had reached the molecular machinery inside the cell. An idea that William James could only describe as plasticity was becoming experimentally accessible biology. Scientists could now ask not merely whether the nervous system changed, but how those changes were created and maintained. Backhand index pointing right. What did this entire journey reveal about the nervous system itself? Look at how far the question had traveled. More than 2,000 years earlier, Aristotle could observe that repeated experience changed behavior. By the 19th century, William James could imagine that habit might depend on the physical plasticity of the nervous system. Then Pavlov brought learning into controlled laboratory experiments. Konorski pushed the investigation toward the organization of neurons. Hebb proposed that repeated patterns of neural activity might change the effectiveness of the connections between them. Bliss and Lømo showed that activity in a neural pathway could produce strengthening that remained after the stimulation ended. And Kandel and his colleagues connected learning itself with changes in synaptic communication. Then followed longer-lasting change into genes, proteins, and structural remodeling. Scientists had moved from observing what experience did to behavior to investigating biological processes capable of preserving the effects of experience inside the nervous system. This capacity for the nervous system to modify its function and, under some conditions, its structure, in response to activity and experience became central to what we now call neuroplasticity. But neuroplasticity wasn't the discovery of one scientist. It wasn't one experiment, and it didn't appear suddenly in modern neuroscience. It emerged from generations of researchers asking progressively deeper versions of the same question. How can experience change us? And now, that question had reached the biology of the nervous system. The story began with something almost anyone could observe. Experience changes us. Practice can change what we are able to do. Repeated experiences can change how we respond. And what we learn can remain with us long after the original experience has passed. For centuries, those changes could be recognized through behavior. Then the investigation moved inward, into conditioned responses, into neurons, into synapses, and eventually, into the molecular processes that help some neural changes persist. What had once been an observation about human experience had become a biological principle. The nervous system is capable of adaptation. But arriving at neuroplasticity did not end the investigation. It created new questions. Which experiences produce lasting change? Why do some changes disappear while others remain? How much can the nervous system change at different stages of life? And what determines whether a particular change helps us or works against us? Those questions continue to shape neuroscience today. More than 2,000 years after people first tried to understand how experience changes behavior, Scientists are still investigating the same extraordinary capacity, not whether experience can change the nervous system, but how, when, where, and for how long. And perhaps that is the larger lesson of this history. Adaptation isn't a single event. It is something living systems have the capacity to keep doing. Throughout life, adaptation is survival,